Now we're moving on to the 5th chapter, which is all about catalysts and enzymes. So start from enzymes. Enzymes are proteins. Protein make enzymes as well. And they are, first we should talk about energy, yeah. So now we're going to talk about biology, energy and biology. Kinetic energy from movement, thermal energy from heat, movement of molecules, speed up a reaction by thermal energy. Potential energy is the chemical potential energy released within these, chemical bonds usually. These potential energy are also, due to location or height or structures. But mostly we're talking about chemical energy. And the activation energy is the energy required for a reaction to happen, which is going up. And enzymes or catalysts are able to make these EA lower. So by lowering that catalytic energy, it will be faster. We all care about how fast a reaction is. Where do we start? I'm kind of lost here. Yeah, so the time is the key factor, and instead of thermic energy, we say gonic energy. So exogonic, endogonic energy. So yeah, catalysts. Yeah, so G is free energy. G is the amount of energy that's required or that is absorbed or that is taken. It's very similar to what we learned in chemistry, just going up and down instead of the thermal energy we talk about now. We talk about gonic energies. Spontaneous is when it's exergonic, so it goes to a lower level, more favorable. When it climbs a level, it's non-spontaneous, which is endergonic, and therefore it's a positive delta G. Negative delta G when it's spontaneous, and they still need catalysis. This one definitely needs catalysis, but this one also needs catalysis, which is exergonic. They do require enzymes to make it at a faster rate. Although it's spontaneous, spontaneous means it will happen naturally by itself, but it doesn't mean it's fast. For example, cellular respiration is a type of exergonic reaction. Cellular respiration is exergonic since they release energy. But photosynthesis is requiring energy, so it is an endergonic reaction. So exergonic for cellular respiration, photosynthesis is endergonic. So yeah, negative and positive G, negative for if it's going down. And this one goes back to normal chemistry stuff, but forming bond will release energy, and breaking bonds require energy. So energy coupling is all about one that releases energy and the other one that will be taking this energy to do other work. That's all it's about. So one will release energy. Exergonic process drives a endergonic process, and these are also referred to by the ATP. So ADP turn into ADP that is being released can be taken by another one to form to react to do the reaction. So ATPs are really useful, really talked about in the energies for bio. So enzymes are proteins, but can also be RNA, behave like chemicals. So enzymes are very selective. They're very specific for certain, usually a few, but substrates. And substrates are one that can bind on enzyme, and this is activation sites. They do not change the delta G, so they do not change the height of which they go down or which they go up, but they do make the energy activation lower. So they lower the activation energy barrier for the reaction to happen. Extremely selective. Enzymes are very specific and selective. So this activation site is determined by composed of amino acid R group so amino acid R group Activation site depends on the R group of the amino acids and temporary bind with substrate. So it has to do with R group in the middle right there. And the enzymes are very specific, which means only specific substrate can bind to yeah, so the induced fit. Induced fit is all about how it is flexible and it is able to change its final shape depending on the substrate. So substrate play a role in determining the final shape of the enzyme, and enzymes are flexible. It's to enhance the ability to catalyze that they are flexible, they are more perfectly fitting to the substrate. Yeah, but it only happens to proper substrates. So when they bind together like this, it's called the enzyme substrate complex. It's just this bound situation right here that's called a complex, and they break down to form what's called products. These are going to be called products. That's all about enzymes. I'm losing my voice making like 7 capsules of these videos already. Reaction rate is the speed of these reactions by the enzymes and stuff. So temperature will look like this. The reaction rate go up, go up, go up. There's an optimal temperature, which is around 37, and they will denature. Denature meaning they die, basically. They will no longer function because they die. The pHs, it will look more like a bell curve. There's an optimal pH, and the further it goes from their pH, well, the lower the rate, and initially, finally, to 0 because they're just non-functional. They're not working anymore. The influence could be temperature, pH, enzyme, substrate, inhibitors, and something I definitely need to note here is that if it is an enzyme concentration, it will look like this. If the substrate concentration will look like this. And that's because eventually this enzyme will also plateau, but that is very unlikely because there's a lot of substrate in real life, especially, and contrarily, if this was about the substrate concentration, well, then we'll plateau faster because there's definitely a limited number of enzymes. So no matter how much substrate you add, if there is only a limited number of enzymes, which is the case, enzymes are a lot fewer than substrate, then they will plateau faster because the reaction rate will at one point stay the same. So it will reach the maximum and stay the same. There's something called initial reaction rate. Not sure what initial reaction rate is. Okay, so now we're going to talk about the graphs. So there is the substrate concentration, initial reaction rate of a function, and the maximum rate of a function. So maximum rate is, of course, this one right here, right? And the Km is the concentration of substrates where the velocity, the speed, is at exactly half. This is called the Michaelis-Menten constant and reflects on the affinity of the enzyme and the substrate. The lower the Km, the stronger the association between the enzyme and substrate. And that's logic because if it's better binding, so if it has a better affinity between the enzyme and the substrate, well, then the reaction is much faster. That's why the Km will be a lot smaller. So they need less concentration for the reaction to reach their half of maximum velocity. And if Km is bigger, that means there's more required concentration for the reaction to reach the half of its maximum, meaning the substrate and the enzymes are better fitting, so they have better affinity. Now talking about molecules that attach to enzymes. Yeah, so molecules that attach to enzymes, there are activators and inhibitors. Activators are the ones that will open up the enzyme for them to activate. And inhibitors are the ones that will make them stop working. And there is competitive and non-competitive inhibitors, where competitive ones will compete with these substrates to try to fit on the activation site. So the activation site. Whereas non-competitive ones will just bind on their allosteric site. So allosteric site is the place where activator and inhibitors will bind. but the activation site is where the normally substrate will bind. Sometimes inhibitors that are competitive will try to bind onto the activation site, making the substrate not able to react anymore. So that is a problem. It could be a problem. So this is all about changing shapes. And cofactor is a metal ion or an organic molecule that will bind to the enzyme and help the function. Most of the time they all need a cofactor. And yep, that's all for